Hi guys, this is Mrs. Hallman, and this is the screencast that's an introduction to our nervous system study anatomy. We're today going to cover the structure of neurons, the organization of the nervous system, and at the end we're going to check out some pretty cool reflex arcs that we have as humans. If you are in my anatomy course, you should have the PowerPoint or should have access to the PowerPoint. If you're not in an anatomy course here at our school, you can feel free to contact me through YouTube and request access. So we're going to get started. So two important things to think about as we jump into the nervous system. When we think about the nervous system, the main key player, the main cell that we focus around is the neuron. And we've talked about the neuron and a little bit about its structure in our histology unit. The two things to remember about the nervous system is first, that with neurons, when they communicate from one to another, it's kind of like a one-way street. Neurons will only ever communicate directions in a one-way flow. So for example, these two neurons or three neurons that we see on our screen right now, we can see that there's one neuron here, two neurons here, third neuron here, and we can see that the flow of information is heading in this direction. There will never ever be a flow of information going the opposite direction because of the physiology and the anatomy of how a neuron works, and we'll learn about that in a second. The second thing to realize is that every neuron will communicate on this pathway in a sequence that will always go chemical signal to electrical signal. So what do I mean by that? Well, when we have one neuron, the chemical signal that's actually sent through the neuron is a signal that will go the whole way through the axon in a one-directional way. The chemical signal will come whenever one neuron will interact with a second neuron. So the chemical signal has to do with neurotransmitters that are released at the synaptic terminal, okay? So if you think about it, we have a electrical signal that goes through the neuron, and then we have a chemical signal that connects it to the next neuron, and then an electrical signal that will carry that on, and then a chemical signal that connects it to the next neuron. So really the sequence is that it will go chemical, electrical, the whole way until it reaches its final destination. So let's talk a little bit about the structure of this cell. Again, we've had a brief introduction to it, but I wanted to make sure that we're all okay with that. When we look at the structure of a neuron, this is a very typical neuron, but understand that neurons actually have different shapes. Typically, when you see a neuron, you see that this end over here is going to have a big cell body, so that wider edge, that wider end that is a little bit bigger is going to be the cell body. So inside of there are going to be all of our organelles that you think about in terms of what we need to do normal cell functions like create proteins and distribute nutrients and to get rid of wastes. Sticking out from that cell body, we have these appendages that are going to collect signals from neurons in the area. Those appendages are called dendrites. And their main jobs, just like branches on a tree, are to kind of branch out and make connections with other neurons. We'll learn the more connections you have in your brain, the more complicated signals and more complicated network that you have that will help you think and make relationships between concepts. In the center of the cell body, this egg yolk-looking structure is going to be the nucleus of that cell body. So moving down towards where the cell body narrows, this area here where the cell body kind of tapers and looks like a funnel, it's actually called the axon hillock, and it will go into this long part of the neuron called the axon. Notice around the axons we have these orange-looking things. That's actually a cell that is flat like a pancake but has been wound around the axon. So we'll talk about these things called myelin sheaths later in this screencast. For now, just know that they are wrapped around the neuron. So technically the neuron is one cell, and the myelin sheaths are actually, in this picture, three cells that are wrapped around the axon of a neuron. So that leaves us to this very end part here. This ending is called the axon ending. I'm going to say that there's probably like 5 different things you could say for these, the end part of the neuron. So some people say axon ending. Some people will say axon terminal, terminal meaning ending. Some people say synaptic terminal because that's where we're going to touch another neuron, and wherever one neuron touches another is going to be called a synapse. Some people call them the terminal buttons, which I think is a cute term. But essentially, this is the basic structure that we'll use moving forward. We said this before, but just remember that the directional signal of a neuron is always going to go from the cell body, from the dendrites really, through the cell body, down the axon, and out the synaptic terminals. Okay? So let's dive into what those myelin sheaths are actually made out of. So here we have another picture of a neuron. We have very similar terminology on how everything is named. But when we look at these myelin sheaths we talked about before, we can see now that we're giving them a second name. So these myelin sheaths are made out of these cells called Schwann cells. They're named after the scientists that first discovered them. And we can see that the Schwann cells are basically flat pancake-like cells that will wrap around the axon. And in between those Schwann cells, we have little gaps where we can see the axon is kind of peeking through. Those little gaps are called the nodes of Ranvier. And I have heard that pronounced two different ways. I apologize if somebody speaks French, and I butchered that. I call it nodes of Ranvier. The myelin sheaths are actually really densely filled with lipids. And if we know anything about lipids, we know that typically they have the appearance of like being lighter in color, a lot of times white. So a fun fact is that those myelin sheaths make up the white matter of your brain. So if you've ever heard of white matter and gray matter, the white matter is actually the area of your brain that has more axons, and the gray matter is going to be the area of your brain that has the cell bodies of those neurons and the axon terminals of your neurons. So what does that look like? Well, when we look at the brain, and if we would take a frontal or a coronal section of the brain and slice it in half and then look at it here, you can see that the outside folds, the sulci of our brain, are going to actually be gray matter. So again, that's going to be where the cell bodies and the synaptic terminals are. The inside part of our brain is going to be white matter. So again, that's going to be where most of the axon terminals are going to, or sorry, the axon parts of the neuron are going to be with those myelin sheaths that give it the appearance of being white. Now, interestingly, as soon as you leave the brain and you head south down the spinal cord, the arrangement of the neurons actually flip-flops and it goes inverse. So in your spinal cord, this is showing a transverse section of your spinal cord. You can see it kind of makes this characteristic butterfly shape, and the outside part of your spinal cord is white, inside part is gray. This is really cool. When we dissect later this year, we'll be able to extract the spinal cord of your pig, and you can actually do a cross section, and if we're looking at some really good preserved pigs, you can actually see some of this pattern there. Okay, so let's talk about how we actually communicate via these neuron cells. So here we're looking at quite a different view of the neuron. We're looking kind of at a cross section of the axon. So the way this works is, remember, the signal that's traveling down the axon is an electrical signal. So what do we mean by electrical if we're talking about cells? Well, electricity is simply just the flow of charges, or specifically the flow of electrons. In biochemistry, when we talk about electrical signals, we're really talking about charged particles or overall gradients of charges moving a signal. So the way that that works in the nervous system is that the cell body is going to be the start of where that signal comes from. And the cell body is going to get that from another neuron that is connected to the dendrites of the cell body. Once that signal collects in the cell body, it's going to start sending a depolarization event. What do I mean by depolarizing? Well, we're aware that polarizing means opposite ends, right? So think about water being polar, or south and north pole. Depolarizing means that we're going to bring the charge a little bit more towards what we call threshold, meaning that there's going to be the start of an action potential. So we're going to send a signal down the axon. The benefit of these Schwann cells is that we said that they're filled mostly with lipids. If you know anything about electricity, you'll know that the power lines on your street where you live are not just going to zap you if you go up and touch them. Now again, don't touch the power lines, but We insulate power lines so that it helps the electricity flow quicker, also so that any bird or squirrel that lands on the power lines is not going to get electrocuted. So in the same way that we insulate our power lines, we're going to insulate our neuron, specifically the axon. And what we're going to do is insulate it so that that electrical communication is actually going to hop from the little areas of exposed axon the whole way down to the end part of the axon terminal. So you can see in this cross section, we have a myelin sheath that's kind of light blue in color, and the charge, that negative charge, is going to actually jump from one side of that node of Ranvier to the next side. We call this entire process saltatory conduction. Why do we need it? Why do we go through the process? Well, saltatory conduction helps us speed up a nerve impulse. And this is pretty important because if you have ever had somebody, like maybe a friend, flick a rubber band towards your face and you flinch, or maybe you hear a firework that you weren't expecting and you flinch, or maybe you put your hand on a hot burner and you flinch. We need our neurons to be extremely quick. Every motion of your body is controlled by that neural network, and we need these communications to be quicker than quick. So we need every help we can in that process to make the communication go really, really speedily. So if we look at what that actual action potential looks like biochemistry-wise, we can call that action potential a nerve impulse, an action potential, or depolarization. We're not going to get too deep into how this actually happens. There's some really beautiful biochemistry that involves protein pumps and action potentials and electrical gradients that if you take AP Bio, we get into like the gnarly detail of that, and it's really quite beautiful. For this level, we're just going to know that there's a flow of ions from one side of the axon to the next. So we can see in this diagram to the right that we can see the progression of time through the first, second, and third axon moving down the page. And we can see that beginning with this first axon, we have a negative charge on the outside of the membrane and a positive charge inside. And then we can see that that blue area where the positive charge is, when we move to the second axon, it actually slides down a little bit. And behind it, we actually flip the charges with the potassium leaving that cell membrane. And we have a negative charge on the inside, positive charge on the outside. And if we look at the third ion, it looks like it's kind of moving down the line a little bit more. So this is a very, very simplified version of what's happening. Basically, we flip the charges on the cell membrane, and that helps us communicate the signal towards that end part of the neuron, or the axon terminal. Okay, so it's important to realize that there are two different types of neurotransmitters. Okay, and we've talked about neurotransmitters before. In anatomy, we talked about one specific type of neurotransmitter in our muscular system called acetylcholine. And acetylcholine, we learned, will directly communicate with a muscle, and it will activate a response on that sarcolemma that will then set off a chain reaction, which will end up with a power stroke of actin and myosin and shortening that sarcomere and causing a muscle contraction. Now, that's one type of neurotransmitters. There are other neurotransmitters that will have other effects. Specifically, we say some neurotransmitters are going to excite and some neurotransmitters are going to inhibit. Basically, you can think inciting is going to flip a switch to the on position. Inhibiting means we're going to flip a switch to the off position. So we can see that in this image, we have one full neuron connecting with a second neuron. We call the area that they connect that synapse. Okay, so at that synapse, we're going to release neurotransmitters, and that's going to be where we communicate that signal. So we look at this image, there's a whole lot going on. So let's kind of simplify things and look just first at this synapse that is shown kind of in the center of the screen. So let's look at this synapse right here. If we blow the synapse up, we can actually see that what's happening is we have this synaptic terminal from one of the neurons in a previous neuron, and we can see that it is connecting with this cell body of this second neuron. So we can say this is neuron number 1, this is neuron number 2. Remember that in the synaptic terminal of neuron number 1, there's going to be an electrical signal that comes down this axon, and it's going to trigger the release of these. These are called our synaptic vesicles. And the green dots that are inside of these synaptic vesicles are going to be our neurotransmitters. The neurotransmitters, when they are released, it involves a calcium ion being flowing into that axon terminal that allows the synaptic vesicles to exocytose to the outside. We call this space in between axon 1 and axon 2, we call it the synaptic cleft. And on the axon body, on the neuron body of axon number 2, we have these little wrench-like receptors that are on that cell body. Those receptors are going to receive the neurotransmitter, and once that neurotransmitter is received, it will then communicate the rest of that electrical signal on to the next neuron. So here's a perfect example of where we can see that we had first an electrical impulse here. It was conveyed to the next neuron using a chemical signal in the form of neurotransmitters, and then we had an electrical signal carry that the whole way through the rest of that neuron's axon. Okay, so let's make sure that we're okay with some of the notes that we see in this overall picture. The first thing is that neurons do not always communicate in a one-to-one communication, meaning it's not always one neuron communicating with another neuron in a straight line. A lot of times we see, and hopefully we see, that in your brain it's a network. So you can see that in this one neuron, so if we're looking at this neuron here in the center of our screen, we see one neuron right here connecting. I see another neuron here connecting, so that's 2. I see another neuron here connecting, that's 3. I see another one here, that's probably 4. There are examples in our brain where one neuron can be connected to hundreds of other neurons. And that's great because that's how we connect ideas in our brain, and that's what makes the function of our brain so robust and so beautiful. So again, all of those neurons kind of branch off from each other, and it means that we can spread a signal and we can connect ideas really, really quickly. It's kind of a beautiful process. Remember also that the neurotransmitters that are released at any time can either excite or turn on a response, or inhibit, which means turn off a response. And again, if you take AP Bio, we talk about integration and how summation occurs, where we actually decide whether or not to send an excitatory or an inhibitory response. Okay, so let us move on to how we actually set up our nervous system. Before we do, this is a good spot just to kind of check in and make sure that you're okay with terminology before we move too much further. So if you'd like to pause your video, see if you can go through numbers 1 through 6 and see if you can confidently label the parts of the neuron now. So go ahead and pause. Awesome. So let's see how you did, and let's kind of go through and label these together. So if we look at number 1, these would be the dendrites. Remember, they are receiving the signals from that network of other neurons that are connecting. Part 2 would be the nucleus. That's that center part of the cell body, which would be number 3. How you doing? Hopefully you got those 3 right. And then we jump on to number 4. Now number 4, if you look really closely, it's actually pointing to this part of the neuron. So that is going to be, and you can put 2 things for this. You could put myelin sheath, or you could put Schwann cell. Excuse my handwriting. Then if we look at number 9, this is the long cylindrical part of the neuron. So this is called our axon. And then 6, there's a bunch of different terms you could put. I'm going to put axon terminal. You could put synaptic terminal, axon buttons, synaptic buttons, anything you really want, as long as you understand the part and its function. So let's move on and start talking about what that actual synapse looks like. Okay, so this is one connection of how the neuron connects with a second neuron. So we can see here that this is going to be our neuron number 1. This is going to be our neuron number 2. We can see again a lot of that same anatomy we talked about before. Here we're going to have that electrical impulse carried from the first neuron. We call this the presynaptic neuron. Let me change colors so that's a little bit more clear. We call that the presynaptic neuron, meaning before. So this one is the pre, okay? Which means that this neuron over here is going to be our postsynaptic neuron. So it's going to be our post, okay? So we have our electrical signal that is going to be transferred from a separate neuron. That's going to trigger the release of these synaptic vesicles. They're going to bind to the outside edge of the synaptic terminal. They're going to exocytose or dump out the neurotransmitters that they're holding. The neurotransmitters will travel across this area called the synaptic cleft, and they will land on special little receptors on the other neuron, the postsynaptic neuron's cell body or cell membrane. Notice too that these receptors look different. So we'll have many different types of receptors on the outside of our neurons, and we'll have a lot of times a different receptor for each neurotransmitter that they were trying to target or trying to send or communicate a signal for. So with that in mind, when the neurotransmitter will bind to the receptors in the postsynaptic neuron, that's going to then transmit that chemical signal back into an electrical signal, and that will continue on to the next neuron, and it will continue on down the line. Okay, so it's important to realize too, we talked about a neurotransmitter in our muscle unit called acetylcholine, and you can see that that one is listed here. Acetylcholine is going to be a neurotransmitter that is specifically used to transmit a signal for the peripheral nervous system in order to kind of make muscle contractions to occur. The other neurotransmitters we have, and again, you don't need to have these memorized, but it's just important that you see that there are both excitatory and inhibitory neurotransmitters. So let's highlight a couple of them here. We have some pretty common ones here. So if you've ever heard of dopamine, this is typically an excitatory neurotransmitter. We talk about dopamine a lot as we go through brain function and we start talking about more psychology. We also have serotonin. So this one's pretty important for sleeping. So this is usually an inhibitory, so this would have a negative response. Norepinephrine is going to be excitatory or inhibitory. And if we keep going down here, we have some lesser known neurotransmitters. We have GABA, which is going to be inhibitory, so it's going to be a negative effect. Glycine, glutamate, aspartate. We'll talk about these a little bit later on in other lectures. And then we have these other ones that are classified as neuropeptides. So those are more protein bases. These were amino acids. These are biogenic amines, and acetylcholine is kind of off by itself. So again, don't have to have those memorized, but just a good idea to kind of have some hip pocket examples of neurotransmitters. So we've talked about the kind of the cell level of how our nervous system works. So we talked about the neurons and how they communicate. Let's talk about the overall system of the nervous system and how it's organized. So the nervous system is broken down into two main parts. We can see in purple on the left-hand side, we have our central nervous system. Central nervous system, very simplistic. It is only composed of our brain and our spinal cord. The peripheral nervous system, you can kind of get a hint of that because peripheral means edges or outside. Peripheral nervous system is going to be pretty much everything except for the brain and spinal cord. So you can see in this image to the right, it kind of is all of the nerves that are going to go from the central nervous system to every other inch of our body. So the central nervous system is pretty simplistic. We'll talk about the brain and the functions of the brain later in class and talk about the spinal cord as well. So let's focus on the peripheral nervous system and how that is divided. So if we just look at the first breakdown of the peripheral nervous system, we have two branches. The first branch is called the autonomic, and the second branch is called the somatic. A really good way to remember autonomic versus somatic is I like to remember autonomic sounds like automatic, in that we typically don't have a whole lot of voluntary control in our autonomic nervous system. It is going to control the parts of our body that happen automatically. So thankfully, you don't have to sit here as you're watching this lecture and remind yourself every couple of seconds to breathe. Your autonomic nervous system will automatically communicate the necessity to breathe. It will also keep that heart beating. Thankfully, we don't have to, throughout every moment of the day and throughout the sleep as well, remember to beat our heart or to blink our eyes or to digest the food that we just drank or to contract the bladder to hold in the urine so that we don't pee ourselves. So those are all autonomic nervous systems, nervous system control. Somatic nervous system, a good way to remember this is that somatic means body. So this really is whenever we're communicating either with our muscles or with our sense organs. This is under a lot more voluntary control. So you can remember this is the rest of the body. So this is how we move our muscles. This is how we choose to breathe in a deep breath and smell something. This is how we control our vision to look in one direction or the other. Okay, so this is a good reminder that somatic means body. Now the somatic nervous system we can see in this slide is broken down further into two other systems. This is pretty easy because really it just is information coming in and information coming out. So our somatic system is broken down into what we call our afferent or our efferent. Okay, so our afferent signals are going to be anything that will go to our brain. And our efferent signals are going to be part of our motor control, so our muscles, something that's going to do an action, our motor output. And those are our efferent signals. They're going to move away from our nervous system. So I always remember the difference between affect and effect. Effect is a noun, so it's the effect that you're going to have. So if I am affected by a really gross smell, the effect that it's going to have is that I'm going to move away from that smell. So it's a way that you can kind of remember afferent versus efferent. Afferent to the brain, efferent away from the brain. Okay. So our autonomic nervous system is also broken down into two different categories. So somatic was broken down into sensory and motor. Autonomic is broken down into sympathetic and parasympathetic. And these two really contradict each other pretty much every moment of the day. I like to think one is kind of like the chill system and one is the excitatory system. So at any point in the day, your body is either amping itself up or trying to relax. So let's talk first about that amping up system. So our sympathetic nervous system is known as our fight or flight nervous system. This nervous system is going to basically excite certain organs, certain muscles, and get ready to either fight an attacker or to flight so we can get away from something. So we call it our arousing nervous system. The parasympathetic nervous system is our calming system. So that's going to be the system that after you kind of excite yourself, this is going to calm you back down. So at any point in the day, these systems are kind of fighting against each other. And the cool thing is that we now have ways through like meditation and through mindfulness that we can really dive in and activate that parasympathetic nervous system. So while both of these systems are outside of what we call our voluntary control, because they're part of that autonomic nervous system, there are ways that we can affect these nervous systems through the actions that we take, through the exercise that we choose to do, and through the mindfulness that we choose to practice. Okay. One little memorization technique that I use, and I don't know if it's going to make any sense to you, is I remember parasympathetic is kind of sounds like somebody saying like, there. I remember somebody like tapping somebody lovingly on the shoulder, like trying to get them to calm down, saying para, be calm, like para. So that helps me remember parasympathetic is chill and sympathetic is going to be arousing. So if we look at this, this image, I realize now it's a little bit pixelated for you guys. Could you label which of these systems that you have control over? So if we go down through these, you can pause. You can see the first one is peripheral nervous system. Then it branches into somatic and autonomic, and then sympathetic and parasympathetic. Enteric is just a further division of the sympathetic and parasympathetic that just has to do with digestions and your intestines. So you can kind of ignore that for now. So go ahead and pause and maybe go through and try to label which ones of these are under your control or which ones of these are not under your control. Awesome. So if you paused and you figured that out, let's go through and kind of check. So our peripheral nervous system is divided into somatic and autonomic. So somatic, remember, is the only one that we do control. Autonomic are under non-voluntary controls, which means technically sympathetic and parasympathetic are also not under voluntary control. So to answer that is really just the somatic. And if we remember, the somatic is broken down into two different categories: sensory and motor. So really, these would be the ones we could control, and the rest of the autonomic would not be ones that are underneath our control. Okay, so let's take a look at how these nervous systems look physiologically. Let's take a look at how the neurons work in these nervous systems. So on the top of your screen, you can see an example of the spinal cord, which is going to have a nerve that's going to, or one axon, or one neuron that's going to go the whole way down here. We can see our myelinated sheaths, our myelinated axon here. So this is an example of the somatic nervous system. And if we do a quick check, remember somatic, is it voluntary or involuntary? Somatic is going to be voluntary. We have a choice. So really, this signal might be our brain sending an electrical signal down the axon and then releasing a chemical signal, aka our favorite neurotransmitter, acetylcholine, to take this muscle and shorten it, make it smaller. Okay, so that would be a voluntary control of the somatic nervous system. If we compare that to now an autonomic reaction, we can see that there are some differences. In our somatic nervous system, it really was one neuron that connected to a muscle, and we would have that voluntary control. With our autonomic nervous system, a lot of times it's a little bit more complicated. Here we have one neuron that is myelinated that's going to release acetylcholine to a second neuron, right? And then that neuron may release acetylcholine, but it also might release NE, which is norepinephrine. We can see that that norepinephrine or acetylcholine might affect a smooth muscle, that spindle-shaped cell we have here. It might affect adipose tissue, or it might affect the branching striated cardiac tissue that we have in our body. So really, the autonomic nervous system is going to affect a greater number of cells, whereas the somatic nervous system is really just going to affect our muscle cells and our sensory cells coming in. Okay. Now, this basically says the exact same thing, but we can see there's a little bit more detail in this slide. So you can see over here, here's our somatic nervous system, here is our autonomic nervous system. The autonomic nervous system is broken down into parasympathetic, that's our chill system, and our sympathetic, which is going to be our excitatory system. So our somatic system, if we look at the top part of our screen here, is going to be a little bit more simplistic. We're going to have a one-way flow of information down a neuron. We're typically going to be using acetylcholine. This is probably going to be our CNS here, so our central nervous system, converting that into our PNS, or our peripheral nervous system. And it's going to affect muscles, and it's going to be mostly stimulatory, so excitatory. In our sympathetic nervous system, so now we're in the autonomic realm, this is involuntary motion. We might have many different ways to transmit that signal. So here we have an example of a one-way signal from a neuron. Then they're going to meet in this area wherever neurons meet in a bundle. It can be called a ganglion. And then that is going to affect norepinephrine, which is going to communicate with the smooth muscle in our intestines. Maybe it's going to stimulate digestion. Okay, so maybe that's a positive signal. Here we have another example of a neuron, which is going to directly, instead of attached to a neuron like it did here, it's going to directly attach to a gland. So a lot of times our parasympathetic nervous system will, our sympathetic or parasympathetic nervous system will not just connect to other neurons or to a muscle, it'll connect to a gland. So the gland that we're looking at here is actually a gland that's called the adrenal gland. And this gland sits right on top of your kidneys. The adrenal gland, especially in the medulla part of the adrenal gland, is going to secrete epinephrine and norepinephrine. So these are two hormones that are going to have the effect of basically transmitting a signal through your endocrine system that will cause a bunch of different results. So that epinephrine would travel through your bloodstream, and maybe it would excite your heart rate, or maybe it would decrease the pancreatic enzyme, the enzymes that are created in your pancreas that would help with digestion. Maybe it would slow down digestion and increase your heart rate. Okay. Now, for parasympathetic, we can have also acetylcholine being used between one neuron to a second neuron, and maybe that parasympathetic system response, if we're trying to kind of calm things down, maybe that would remove that accelerated heart rate, slow down your heart rate, maybe increase digestion, increase the peristalsis, that smooth muscle contraction that helps us with digestion. So basically, all of this is to show you that in your nervous systems, there are different ways that we can communicate, not just neuron to neuron. Okay, so this slide does a really nice job of showing you the differences between our sympathetic on this side and our parasympathetic on this side. So remember that our parasympathetic is going to be our relaxed. So this is going to be kind of our chill nervous system. And this is going to be our excited nervous system. Okay, so I really like this image because it shows you all of the different ways that these two nervous systems can affect our body. So you can see if we just kind of start on this half of the screen, when your sympathetic nervous system gets activated, it will dilate your pupils. So this is actually going to allow more light to enter your eye, and it's going to increase temporarily your ability to see movement. This is a good adaptation because as we have evolved, we have been from time to time prey species, and we need to know when it is that there's a big mountain lion about to attack us. It will inhibit salivation. Why would it do that? Well, if you are running away from a polar bear, most likely you're not worried about chewing and swallowing food. You're worrying about getting away from that polar bear. So we don't need a whole lot of saliva to help us with that digestion. It will dilate our bronchiole. Remember, dilate means to get bigger. So if our bronchioles are normally this size, dilation will make them be this size. Why is that important? Well, your bronchioles are going to help you get more oxygen to and from your lungs, or more gas exchange in your lungs. So remember, you're running away from that mountain lion. You need to have more oxygen to your muscles so that you can run faster. It's going to inhibit digestion. So again, we don't want to send blood to your stomach to digest what you just ate. We want to send all of the blood that we have to your muscles so you can continue to run away. It's going to stimulate glucose release by your liver. If you remember from bio class, your liver will store glucose in the form of glycogen, and we need a lot of energy to move the muscles to run away from the mountain lion. So we'll actually trigger the liver to let go of that glucose so that we can run. It's going to release epinephrine and norepinephrine. Those are two excitatory hormones that will elevate our heart rate, that will get our blood pumping so that we can run even faster. It's going to activate our sweat gland. A lot of times it will relax the smooth muscles that will go around our bladder. It will contract the rectum. And what that means is that we will literally hold in our poop, but sometimes you pee yourself when you are scared or excited. Both of those are just the sympathetic responses. If we move over here to our chill responses, we kind of have the opposite of all of these. So we're going to constrict our pupils, which is why when you're relaxed, you might not need to be as alert and to see movement as much. We're going to stimulate saliva so that we can start to digest. We don't need as much oxygen, so we're going to decrease the diameter of our bronchioles. We're going to send all that blood that we just had going to our muscles to our stomach. We're going to stimulate our gallbladder because our gallbladder makes a lot of the juices that are needed for digestion. We're going to contract our bladder. We're going to relax our rectum, and we're going to vasodilate. The other reactions that the sympathetic and the sympathetic and parasympathetic nervous system have to do with sexual intercourse. So they have to do with whether or not the penis would become erect or flaccid, or whether or not the different genitalia that are needed for orgasms would excite or inhibit that excitement. Okay, so this does a pretty nice job of showing, again, how the somatic versus the autonomic nervous system will affect in terms of where they are going to go in the brain. So we can see in this image, we have a picture of our brain. This is going to be our SNS, or our somatic nervous system. On this side, we're going to have our autonomic nervous system. So we can see here we have a pretty simplistic pattern. We have parts of our brain associating with different parts of our muscles. This is pretty cool because we have technology where we can simulate a part of the brain, and depending on where we simulate in the brain, it's going to affect a different type of muscle. In our autonomic nervous system, a lot of those effectors, a lot of those neurons will be more central to the brain. So they will be more towards our brain stem, towards our thalamus or hypothalamus. We can see this one, for example, is going to connect and affect our smooth muscle. We can see this one here is going to affect our cardiac muscle. So again, we can see that they travel further down through that brain stem, and they're going to control more of those automatic responses that we have in our body. Okay, so that kind of wraps up the anatomy of the nervous system, the organization of the nervous system. So let's talk about these funny things called reflex and reflex arcs. Up to this point, every incoming response, which again would be an afferent signal, and every efferent response, which would be a signal that goes away, will integrate, or that means it kind of is processed in our brain. Reflexes and reflex arcs are the exception to those rules. So pretty much everything that happens in your body will always be transmitted into your brain. We will process it and then come up with a signal of what we're going to do from the brain to the other part of your body. A reflex is anytime we skip that integration point, or when we think about what we're going to do with that incoming signal. So what that means is a reflex is any time we're going to have an involuntary response to a stimuli. Involuntary in the fact that it's not a choice that we actively make from our brain. A reflex arc is going to be the direct route from an incoming sensory neuron to this thing called an interneuron, which would be in our central nervous system, in our spinal cord, but it does not go to our brain, and then to an effector mechanism. And it's easier to show an example of this to truly understand it. So one of the really famous reflex arcs that we have, I'm sure at one point in your life you've gone to the doctor and they've had you sit on a stool or a table where your leg is freely swinging, and they take a very sturdy mallet, not to hurt you, but they take that mallet and they just tap the very edge of where your patella is, so in between your patella and your tibia. And what that does is it will stimulate a really famous reflex arc called your patellar reflex. And what that does is it will cause your quadricep muscle to contract. The reason why it does is that patellar tendon will have a neuron that senses that pressure from the little mallet. It will travel to your spinal cord, but notice it doesn't go from your spinal cord up to your brain. It loops back around and it will have an efferent nerve carrying a message to your quadricep muscle to contract. And usually what happens is if your foot is just kind of freely floating and the doctor hits the front part of your patellar reflex, your foot will just involuntarily kind of kick out. I'd encourage you guys, if you have a willing subject at home, you can use something heavy, not like a hammer, but you can use maybe like the blunt end of the handle of your scissors and just lightly tap on a family member's front part of their knee. Again, pretty gently, you don't need a whole lot of pressure for this. Make sure their knee is hanging freely and you can actually find that patellar reflex. So it's kind of fun to do. So let's talk about other reflexes. So there are reflexes that are associated with the sensory of pain. So our body is pretty smart, and we have evolved these pain reflexes. So for example, this is showing that if you would put your hand on a tack or something sharp, or if you got stung, your reflex is always to move away from whatever that stimulus is. So you can see here in this reflex arc, we have step one is that original stimulus. That stimulus would be picked up by a receptor in your skin. And this would be what type of neuron? Well, this would be an afferent neuron. It would communicate it to this little white piece of a neuron here called our interneuron. And then it would communicate it through this efferent neuron, which would be our motor response. And that would then communicate with this part of your muscle in your biceps, which would move your hand up and away from that stimulus. So we have these pain reflex arcs a lot of the places of our body, so that anytime you would unknowingly get stung or get cut by something sharp, our automatic reflex before we even think about it is to move away from that stimulus. Same thing with heat. So a lot of people think about, you know, once in your life, I'm sure you've touched something a little hot, like the edge of a pan, or maybe you've put your hand on a slightly hot stove. This is showing what happens when you put your finger too close to a flame. I don't know why you would do that, but this shows it. Again, we have our sensory neuron here. So that would show us that incoming signal, that afferent signal, that would then go to an interneuron, which would then go to our efferent neuron. So again, this is our afferent. So it's going to our brain. That's our sensory neuron. And then we have our efferent neuron that is going towards our motor effect. And it would usually constrict maybe our extensor digitorum, which would be in the top part of your arm, which would then lift your hand away from whatever that stimulus would be. In this case, it's the burning candle. I don't know why you're touching candles in the first place. So I threw this link in here. This is a quick little video about reflex arcs. If you're interested, we're not going to watch it right now. I wanted to give you some examples of human reflex arcs to kind of leave you with. Really interesting one, sneezing and coughing are both reflex arcs. So we don't think about sneezing all the time. Specifically, if you've ever walked into a room where it's been really dusty, your sensors in the inside part of your nose might receive that you are breathing in a lot of dust or dirt, so they'll trigger you to sneeze to get rid of that. Coughing is the same way. I'm sure even though you guys are really good at eating and drinking, every now and then you take a gulp of water and it goes down the wrong pipe. Coughing is an automatic reflex to that. The knee jerk is that patellar reflex we talked about. Blushing, I think this is really interesting, is actually a reflex as well. So there are people who actually have a condition where that reflex is triggered throughout the day, and it's completely involuntary, so they will blush without thinking about it, and they can't really control it. Blushing typically happens for you if you're embarrassed or if you're surprised, you're startled. It just means the surface of your skin will vasodilate, releasing more blood to the surface of your skin, which makes you look a little flush or pink. Yawning, really interesting. There's like so much we could talk about with yawning. It's so interesting. And we still don't fully even understand why it is that when you see somebody yawn, you yourself want to yawn. But we do know that it's a reflex arc, that it isn't integrated too far into your brain. We do believe it's a reflex. The mammalian diving reflex, I think, is one of the most interesting, and the reason why I have pictures of swimming babies on this slide. So the mammalian diving reflex is a reflex that when your face is exposed to cold water from infancy, your body is triggered to slow your heart rate and to hold your breath. And it makes kind of sense why we evolved this trait. As mammals are young, if they ever were to fall in the water before they're able to really swim or make the conscious choice to like stand up and get out of the water, it's important for survival that they have a reflex not to breathe in water. So this reflex evolved so that as soon as their face touches cold water, they will close their mouth and stop breathing in. The other part of this is that it will send all of the blood towards the core, and it will send all of the oxygen to the core. So that means it'll actually lower the heart rate of that infant so that it will preserve the most vital organs like your heart, your brain, and your most vital organs inside of you. The last reflex is called the post-matriculation convulsion. So matriculation is a really fancy word for peeing. Convulsion is a really fancy word for shaking. And without getting into too much detail, I'm sure that one or two of us have ever had the sensation when you just finish peeing where you get a little shiver running up your spine. It's actually scientifically more experienced in males than it is in females, but that's actually another example of a human reflex. So fun fact there. I want to leave you with two other references. This shows you the mammalian dive reflex. While the mammalian dive reflex is really shown more in infants, we do still retain some of that reflex as adults. So this video is less than 2 minutes long, and it basically shows a man put his head into cold water, and at the same time, he's hooked up to a heart rate monitor. And you can see that as the longer he puts his head in this cold water, his heart rate actually drops, which is a remnant of this mammalian diving reflex. And lastly, on the right-hand side, I included a really fun link that's from a Popular Science article all about the mammalian diving reflex. It's also called the Brady cardiac reflex. So you can read more about that if you're interested in why some people throw infants into pools to see what they do. So all of that concludes the nervous system lecture for this chapter. I hope that was helpful.